Well, thank you so very much. Thank you for the invitation to invite me back, and that you haven't gotten weary of any of my jokes and other things like that. So I'm so thankful for all of that. Okay, we did a three-part series this week, and we started out on Wednesday morning in Romans chapter 1, verses 18 through 25. We took it apart, verse by verse, point by point, and we found out that there is a truth that is revealed, and the revelation of that truth is ultimately that you can see God's invisible attributes, and you can know that He is the agent. He is the agent in creating the universe. He is the agent in creating nature. And we recognize His agency by His what? Handiwork or workmanship. And there are distinctive features of that, and then we see it. And that there is a suppression of that truth that is very offensive to God, and His wrath is revealed against it. And ultimately, the suppression of the truth exchanges the truth of God for a lie. And it exchanges the fact that He is the agent who creates nature, and they eventually come up with the conclusion, the erroneous conclusion, that nature creates itself. Nature creates itself. And Dr. Gunn yesterday gave me a good point and a good tip as we were talking last night. Grammatically, only really living things, and particularly living things with intelligence and volition, can be an agent. Inanimate things can be an instrument. And so we see a real confusion on the part of these people where they say that nature somehow in and of itself can exercise agency. And then we see that the manifestation of the belief that nature can exercise agency is through Darwin's really pretty clever mechanism of how organisms supposedly adjust and adapt, and that is through what? Natural selection. And the key word there is selection, because selection indicates the ability to exercise volition. In other words, to be an agent. And it is a mystical mental construct. And this is a good way for you to remember it. It's a mystical mental construct. Nobody has ever seen nature select anything. It is never selected. So it's always a metaphor. And it's a metaphor where people are projecting on to nature selective ability in place of God's selective ability. And when you project, Dr. Ice just mentioned it several times with Israel, when you project onto an inanimate thing, like a stone or a carved piece of wood, godlike or even human-like powers, what is that called? It's idolatry. And people used to manifest it in a statue or something. Now, we're just more clever. We just project it onto what in general? Nature in general. And the view is always that nature is shaping and molding and making organisms as it drives them through space and time. And organisms are essentially like modeling clay, being molded and made by their environment. And we're arguing that they have it 180 degrees wrong, that organisms are not these passive things being driven through space and time, but organisms are active, I'm giving you all the key words here, active, problem-solving entities that drive themselves through space and time and solve all these different problems. And they have it because they have ability to sense their conditions. They have logic built in the inside that says if there's this condition, then do this. If this condition, then do that. And they have output mechanisms. So today, we're picking up, and I'm going to present a kind of a scientific paper that I presented last year for the Creation Biology and the Creation Research Societies. We're going to go through, not the entire paper, we're going to go through a good portion of this paper. I'm going to give you ways in which organisms fill the earth. And that was the key word. In Genesis chapter 1, verse 20, organisms were commanded to be fruitful, multiply, and fill the earth. And this command was given before the fall or after the fall. That's an easy one. Before. So it could not have been a death-driven survival of the fittest process. So there had to be mechanisms for organisms to fill a dynamic earth even before there was ever death. And this is what you're going to get from this talk. God's genius and creativity is phenomenal. So we're going to look at many ways in which organisms fill all these different niches, and not a single one of them is going to be driven by death. And they're all going to be, wow, I didn't know, different ways. And we're going to look at it a spectrum from physiological changes, where you change something like right now, if I were to yell, bang, really loud, some of your heart rates just went up. That's a physiological change. That happens in seconds. And then we're going to take it all the way out through multiple generations. We're going to see all these different types of changes. So people will ask you, well, if you're knocking down natural selection, what do you have to replace it? Well, I have a mechanism which I would call continuous environmental tracking. That organisms are able to continuously track along with environmental changes. And it's going to be analogous to a man-made tracking system. And sometimes the organisms are going to track consciously, but more often than not, they're going to track along unconsciously. In other words, organisms are going to operate in a very automated and autonomous way. And I use those two words because I want to push back against some evolutionary thinking. Right now, some of you guys just ate a bagel or something like that, and it's in your gut, and it's being processed in an automated way. You're not thinking about it. I need to digest my bagel. You know, it's grinding it out, and it's digesting it in an automated way. You're breathing in oxygen, and it's going through a long process to have respiration to give you energy. It's automated. And now, I want you to appreciate how hard it is to make fully automated things, so that you appreciate the genius of God. So you operate in a very automated way. You're also autonomous. Now, autonomous does not mean that you are independent from God. Autonomous means this: you are not part of Gaia. Dr. Nicholas is an autonomous individual. Amen. Who will stand accountable before God for himself. Amen to that. I don't want your sins. So he's autonomous. He doesn't want my sins. None of us want Dr. Hawking's. So autonomous. I'm not defined in terms of him. He's not defined in terms of me. We work together very closely as part of a system. But I'm never amalgamated into the system. I don't become one with the collective. And that thinking is out there. So we are automated, autonomous systems. Now, am I still dependent on God for my daily bread? Yes. I need that energy. So even though I'm an autonomous thing, I still need to bring in energy, I still need the food, I still need to pray, give me my daily bread, all of those things. That's what I'm saying. So this is how organisms function, and when you see that, believe me, it will increase your appreciation for the greatness of the Lord. So these are the two questions we've been talking about all week. Does the data indicate that organisms, their form, that is their basic shape, is it externally imposed on them over time by fluctuating environmental pressures? That's what evolutionists say. It's imposed on you. Or do they, like drones, and I'm going to illustrate it with a drone, drive themselves through space, time, continually tracking dynamic environments while making suitable changes to their own forms by internal means. Externally imposed versus internally given, designed into them. Well, evolutionists always have these dynamic fitness landscapes, and they illustrate them as like a hill. And fitness is at the top of the hill, and geology and climate drive diversification. They drive creatures up to the top of the hill, like king of the hill. All the time they're being driven, what's happening to most of the creatures? They're what? Dying. And soon just a few make it to the top of the hill. and then the landscape changes and they get driven over and over and over again. I'm saying, well, maybe the idea of fitness landscapes changing are fine, but instead of being driven to the top of the hill, they drive themselves to the top of the hill. And it probably operates like a drone. Now, a drone, you know, is an unmanned craft. And this is going to be, it's actually been built, a submarine hunter. There's no sailors on board. And it hunts down submarines. And the article says this new vessel is going to patrol the world's oceans, tracking down enemy subs, and will execute its mission without a single person on board. Now, how does it do that? Well, it's packed with exquisite sensors, all kinds of sensors. So if there's anything that sub is putting out or emitting, there's a sensor to find it. You see what I'm saying? Sounds, smells, anything. There's a sensor to find it. And it's full of a bunch of logic and computers on board that's able to track onto that submarine. And supposedly, according to the story, once it locks onto a sub, the sub's a goner. You know, it will hunt it down on that. Well, there's a sophisticated tracking system built into it. I'm going to argue that you and all other creatures have sophisticated tracking systems, enable you to track your environment, that make this submarine look pretty paltry. And they're quite incredible. And we need to start looking at creatures in a very automated way, like this, possibly. Here's the sound. Sound.
Years of daring that we look to offer drivers peace of mind. 50 years of designing cars for crash survival that led us to our most revolutionary feature yet. A car that can see trouble and stop itself to avoid it.
Did you see the car? It's a Subaru. And they launched it unmanned towards the wall, and it did what? It stopped itself. Did they say stopped itself? Yes, they did. They did. They say it can see it, and it stops itself. Now, I point that out because an evolutionist always would say the wall stopped the car. They see everything as externally driven. And in fact, they don't even call things on organisms sensors and detectors very often. They always call them what? Receptors. Because they see organisms as passive. They call them receptors. But I'm saying, no, they're actively surveilling, they're actively detecting, and the car stopped itself. You do the same thing. You're very automated, except you're much more sophisticated than this car. Now, did you see the engineer at the end? He looked kind of what? A little smug, a little proud. Should he be? Yeah, this is a technological achievement. He probably worked hard on it, on that. Well, God should be a little smug, so to speak, at his incredible technological achievements. And we should honor him for that, and we should respect him for that. Well, so what are tracking systems? Well, there's 4 minimum components. And I've researched these. There's sensors, which we all know detect things in our environment. There's condition consequence elements. This is very important. It says when I detect a condition, I will execute a specific consequence, a specific behavior. Now, only the person who knows what the conditions and the consequences can be can build this kind of logic into creatures. There are output responses, which were the same 3 things I talked about yesterday, but for tracking systems, they generally have a little clock on them so that they can actually intercept something at a point in place and time, so they have a chronometer there. It's like sea turtles, which their eggs are laid on a tiny island off the coast of Africa, and then the little hatchlings get into the ocean. They swim all the way across the Atlantic Ocean off the coast of Brazil, and then they turn around a year later, and they find their way back to that island, which is like 7 miles across. That's pretty good tracking in order to hit that island all the way across. Well, here's a man-made tracking system. It has a sensor to sense the sun. This thing is going to follow the sun. So as a sensor to send the sun, if you follow the pathway, it sends it down to some computer some logic center, and then it sends signals out to drive motors, which turn the sensor around and up and down so it stays tracking with the sun. Make sense? That's how it does it. It's able to track. It has all those key components. Well, organisms, believe it or not, have all of these same components, sensing, logic, output, and many of them have a little chronometer, clock built right into them. And this is where we see it. This is from a textbook. This is a blown up picture of a cell membrane. Here's the inside of the cell. And studded all over the cell membrane are, guess what? Tiny little sensors and detectors. And they're able to detect what they call environmental stresses, all the different things in the environment. And in a very complicated way, they send a signal down to the nucleus. And you tend to think that the nucleus, where the DNA is the logic center. And it is. But as you notice, the signal is getting sent through all of these very complex pathways, and they're actually using logic on the signal as it's being sent to the nucleus. So logic starts almost as soon as the signal is picked up. And then the cell responds. And this can be blown up from the cellular level to the organ level to the organism level, even to the ecosystem level. It operates the whole way, all the way through, from cells to organisms to ecosystems. They have sensors, logic centers, and output devices in order to respond. Except they're much more sophisticated because they're microscopic and they're very, very tiny. So we're going to look at 13 different ways in which organisms do this, and we're going to look at them over time. We're going to start right here with FO. That's the parental generation. And when you see 1, this is a science poster, so I'll explain on there. F1 is the first generation. F2 would be the second generation. So we're going to look at some of these things over time. And we're going to look at what in the environment is the target, where the sensor is on their body, what the specified self-adjustment will be, and then just for fun, some principles of design that are going to illustrate the things we're talking about. Some of them are going to look a little complicated. I'll explain them to you. But right here at the physiological level, like this says gut commensal E. coli bacteria, blah, 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 on the topic. But what it's showing you is how the bacteria in your gut relate together. And the target are cells in your gut, enteroendocrine. These are cells in your gut which secrete hormones. But what they're sensing for are changes to the protein output of the E. coli. Now let me show you how this works together. Look at this. It's a picture of a person eating an apple. Food goes into the gut. Your gut's full of these E. coli bacteria. As soon as you eat them, the population of the E. coli goes up. It explodes. But not only does the population change, the proteins that they're making change. They produce different types of protein. And guess what? Certain cells in your gut, these enteroendocrine cells, they have detectors for those proteins. And when they detect those new proteins in the gut, they start making different substances themselves and dump them into your bloodstream. And some of the proteins that the bacteria make can be absorbed directly and put right into your bloodstream. So your gut is looking for the proteins that these bacteria make. And when they detect them, they produce different substances, put them into your bloodstream, they travel up to your brain detects those substances, and it gives you the feeling of being full. It shuts down your appetite. That's pretty incredible. Quite incredible. Because it doesn't just shut down the appetite because you stretched your belly. The bacteria are working what? Incredibly close together with your gut cells to make this so that you quit eating. Most of us say, I wish I had more bacteria on that, on there. But that's how it all works together. Now, some people say we've become one with the bacteria because can you see how close this is working? It's like a hand and a glove. But you don't become one with the bacteria, and the bacteria don't become one with you. Bacteria is still bacteria. You're still you. God made them to work incredibly close together with a bunch of sensors. All right. 2 autonomous entities, in order to get them to work together, must have an interface. You just don't get bacteria and humans say, now play together nicely. You have to have something which makes them work together. And generally what we call the immune system should be called an interface system because it is allowing you to work together with your bacteria or viruses or anything else. You have to have an interface. Those enteroendocrine cells are part of the interface. That is another design mechanism. Okay, flies. Flies travel around and based on their species, they like certain humidity and they like certain temperatures. Now, how in the world do they migrate from temperature to temperature and humidity areas? Well, this paper just published last year found the humidity sensors in these flies. So what are this target? Relative humidity, and they have a sensor for dry, moist, cold, and other things. Now, where did they find this sensor? Well, they found it on its antenna. And here's a picture of where the antenna is. And if you magnify this greatly, down in a tiny little pocket were these little structures, tiny little structures, which nobody knew what they did, and they turned out to be humidity sensors. Incredibly small humidity sensors. And they can detect all kinds of things. And then another little pocket on the antenna, they have a temperature sensor, 2 different sensors. Anyway, they can detect minute changes in humidity, and when the humidity changes, they migrate to the place where it has the humidity that they like. So they're able to detect it. So you see, I'm taking you as sensors on how organisms are filling the earth and how they continually fill the earth and fill different environments. This is the smallest man-made humidity sensor which can do something like it, and this one is microscopic. That one's fitting on a quarter. So it kind of gives you an idea of just how incredibly complex and how sophisticated this tiny little clump of cells are, or structures, in the base of an antenna. Now, ever since I used to not really worry about swatting flies that much, but now every time I swat them, I think, wow, I just destroyed something that's far more sophisticated than an F-16. Crunch, you know, and it's really given me a different appreciation of just what I just wiped out on these things. One of these days we won't, and we'll just appreciate all that the Lord has made. Okay, here's the design principle. Internal programming always specifies which conditions will be a stimulus. I talked about this yesterday. Things outside of you are not just stimulus. There's something inside you which said, the programmer and the designer said, I want this to be a stimulus, and then he equipped you with a what to detect the stimulus? A sensor. So everything is internal, even the specifications. And biologists generally get it wrong. They say, well, look, the humidity's out there. It's obviously driving the flies around by the changes of humidity. Wrong. The humidity is what the humidity is. And the flies find the humidity that they like. Do you see what I'm saying? Completely different way of looking at it. All right, here's another one, a gland. A gland which allows certain birds to live in a saltwater environment or a freshwater environment or go back and forth between the two environments. And it says it's a model study for membrane biogenesis because this gland is sometimes in the bird and sometimes it isn't in the bird. And it will rapidly grow when it needs to. And there's a target of the sodium concentration inside the bird's blood, inside the bird's plasma. So birds are maybe drinking salt water. Their sodium concentration does what? It goes up. They have a sodium detector in their vasculature near their heart, which is constantly monitoring the sodium concentration. So when they migrate to a saltwater environment and their sodium concentration goes up, a signal is sent to a part of their body right here which says, grow this gland. And it grows rapidly. And the gland actually operates a lot like a kidney. And it has loops just like a kidney, and it filters out the sodium and it excretes it out by the nose. And so suddenly this bird now can migrate to a saltwater environment, and it fills the saltwater niche. And if the bird migrates away from the saltwater environment back to freshwater, guess what happens to the gland? It disappears. And it comes back and forth, back and forth. Now, some creationists wrote this up that the salt water triggered the gland. It doesn't trigger the gland. What triggers the gland? If you're following along, it ingests the sodium. It has a sensor which detects the sodium, sends a signal to its own body saying, make the gland. That's how it works. That's because true triggers are always the sensors which are part of the thing, the entity. Nothing outside of you is the trigger. The trigger will always be a part of you. So if I assign you the project of making me a gun, I say, make me a gun. I guarantee this lady, some in time in her design process, is going to put what on the gun? A trigger. She will stick it on the gun and she will specify what she wants to trigger, what she wants that trigger to be tripped by. Will she not? They're always part of the entity. Okay, now we're going to fill a different environment. High altitudes, and this is going to go through multiple generations. Now, if you and I were to go up to the top of Pikes Peak, we would acclimate to the change in altitude starting within a few hours, and then within a few days we would acclimate, and that's because your body has a sensor to detect the partial pressure of oxygen, and when it decreases, you start to produce more red blood cells, so you can carry more oxygen. The trouble is, the more red blood cells you produce, the thicker your blood gets, the viscosity goes up, and if you stayed in that condition for a long time, you would go into heart failure. But Tibetans, they live up at very, very high altitudes, 14, 15, 16,000 feet, and then they go up to the top of Mount Everest without even carrying oxygen. How do they do that? Well, they have acclimated to these high altitudes, and this paper says, through the introgression of Denisovan-like DNA. Now, Denisovans are an extinct group of people, lived probably about the same type of Neanderthals, fully human. All we know them from is some bones that have been discovered, but we've been able to extract their DNA from those bones. Now, what's interesting is we have sensors which can detect the partial pressure of oxygen, and they're in tiny little organelles in your cells called mitochondria, and they're measuring the flux of electrons going across it. Nevertheless, they're pretty exquisite in detecting oxygen. Now, these folks, they don't adapt, they don't self-adjust like we normally do. In fact, they down-regulate the regular process. They don't produce more red blood cells, making their blood thicker, putting them into heart failure. They have other mechanisms where they more efficiently carry the oxygen. And what's interesting is the DNA of these Tibetans are only found in these Tibetans, a small pocket of Chinese, and guess who? Those extinct Denisovans. You don't have it, Neanderthals don't have it, Melanesians don't have it. Only they have it, and the Denisovans have it. Now that's interesting. So the paper says, we now are also starting to understand that adaptation to local environments may have been facilitated by a gene flow from other hominins, that's what they call the Denisovans, that may have already been adapted to. You notice they're passive. Actually, they were already adapted by themselves to those environments. So we know one thing. Since the Denisovans came off the ark, they were some of the offspring of Noah's sons, at least one of them already had genetic information enabling them to be fruitful, multiply, and fill environments, including what kind of environments? High altitude environments. They already had it. They had it so that they could be fruitful, multiply, and fill those environments. And guess what? They went rapid because they found similar things of people who used to live in the Andes in South America at 14,000 feet. Wow. An entity's features, their traits determine what exposure is stressful or not. I take one of you and I plop you up there. That's a stressful environment. I take a Tibetan and I plop him up there. It's not stressful. It is not the environment which determines what's stressful. It is your traits which determine what is stressful. Oh, change the thinking. You take the same environment, you put two different things in it, one responds one way or the other. It's always your traits. It's always your traits. Okay, here's another one. Mom knows best. Well, that's a good one. The universality of maternal microbial transmission. Let me explain this one to you. We fill different environments, we eat different foods, and your gut bacteria help you process them and help you gain nutrients out. Obviously, wherever you go, you eat different types of food, and the gut bacteria are constantly being regulated, and they're changing their numbers and composition so that you fit the environment you're in. A few weeks ago, I'm on the East Coast, and I'm eating East Coast food, and I come out here to California, and I eat California food. Different types of food. Believe it or not, my gut bacteria are adjusting because my so-called immune system is an interface which is regulating the what? The numbers and the types so that I fit my environment. Well, Mom is already fit to her environment, and then she has little baby. She has little baby there. Well, baby, more than likely, is going to be in the same environment as Mom has. Well, most of the time, this is one of the complicated ones. Part of our immune system, these cells, which are the target, are zeroing in on a certain receptor or a certain molecule that is lining her gut. I'll explain it to you here. Inside of Mom's gut, these are gut cells, she has bacteria. There are certain cells of her interface system which are able to engulf the bacteria whole without killing them. It engulfs the bacteria, it doesn't kill it, and normally it migrates to a lymph node which has this molecule on it. So it homes in on the molecule on the lymph node, and it presents the bacteria to the lymph node whole and live. The lymph node analyzes it. It mobilizes other cells in your body to regulate the numbers and composition of the cells in your gut. Isn't that kind of interesting? That's fascinating. Except when Mom is pregnant and she's lactating, that same molecule which is on the lymph node also shows up on her breast tissue. So now that cell engulfs the bacteria from her gut live. It migrates to the lymph node, but it also migrates where? To her breast tissue. Her breast tissue, where it dumps the live contents into breast milk, and then baby drinks the breast milk, taking live bacteria into baby's gut, seeding baby's gut with the bacteria that Mom once had in her gut, making baby fit for that environment. Did I tell you that the Lord's ways are like really creative? They're really creative, incredibly creative. I mean, you should be saying, whoa, who would have thought of that? That's an incredible way to do this. You just took live bacteria from Mom's gut and you just transported and got it into baby. So an incredibly complex way so that baby now can be, where's baby? Be fruitful, multiply, and what? Fill Mom's environment. Wow. The paper said this. The consequences of entero, gut, mammary, breast, trafficking include the release of dendritic cells, those are the cells that engulf the bacteria, containing live maternal gut bacteria and other antibodies specific for the maternal gut bacteria into the colostrum of breast milk. So it's a mechanism that now Mom is using to enable baby to fill her environment. Wow. Now let's talk about three different types of called epigenetic changes. Epigenetic changes are changes which enable organisms to rapidly fill rapidly changing environments, meaning they don't change the sequence of your DNA. The sequence of the DNA is not changed. What is changed is how the DNA is managed. And certain molecules are placed on the DNA which regulate it, saying, express this DNA or don't express this DNA. Or another way to do it is, you remember that DNA will be transcribed into RNA, right? And then RNA will be translated into proteins. Does that bring back any memories from biology? Sometimes the DNA is transcribed into RNA, but the RNA is not translated into a protein. that RNA molecule goes back and reattaches to the DNA, regulating how the DNA is expressed. Those are epigenetic, above the genome changes. So they're not changing the genes, they're changing the way the genes are expressed. It's very, very complicated. Now, I'll just do this one very quickly. This is a paper on paternal intergenerational epigenetic response to heat exposure. Guinea pigs beat climate change by tweaking their own DNA. That's kind of like the layman's title on this. So these guinea pigs, the dads were raised in a hot environment. Not so hot to cook them, but hot enough that it was not normal. And then they were mated with females that were raised in a normal environment. And then they had pups. And guess what? They sequence the pup's DNA, and they also check for epigenetic changes in the pups. And the pups have changes which enable them to live in what kind of an environment? A hot environment. So they are changing their genetic expression, enabling them to be fruitful, multiply, and fill a rapidly changing hot environment. And the changes are specific for heat regulation. Yesterday, I went over this one. Paternal olfactory experience influences behavior of the neural structure. Do you remember this one? It was the mice, the male mice, who were exposed to cherry blossom odor, and they have a detector in their nose for that cherry blossom odor. You remember the experiment? You take the male mice, you put them on a what? Metal pad that shocks their feet. And you expose them to cherry blossom odor, and you shock their feet. Not lethal, but painful. And then you expose, shock, 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 shock. Biologists love that. And then you take this male mice, and you mate him with a naive female. Remember last year I said not the type of naive females that humans are looking for, but a naive female that has never been exposed to cherry blossom odors. Then they have pups, and you sacrifice the pups, and you stain their tissue up in the olfactory region. They have little bulbs, which are sensors. The blue stripes are the nerves leading from those sensors. Here's the control. Here's another one. Here's cherry blossom odors and foot shocks, and voila. In the pups, which have never smelled cherry blossom odor in their life, they have this huge increase in sensors and nerves for cherry blossom odor. Now, what if it wasn't cherry blossom odor? What if it was fox urine odor? Now Dad has been exposed to fox urine odor. He passes on this information to the pups, and they're already prepped for that in their environment. Something like that. Wow, these allow very rapid changes. Here's a really interesting one. These birds. These birds are living in Russia. In the mid-1980s, Russia had a massive problem with one of their nuclear reactors at a town called Chernobyl. And it spewed radiation in very, very large areas all over that site. Very, very high concentrations of radiation. Now you would think that it would be devoid of life, but it isn't devoid of life. In fact, there are lots of living things in that area of radiation, including these birds. These birds are living quite happily in that area of just very, very high dose radiation. Look what it says. Chronic exposure to low dose radiation. Well, it's low dose compared to what it would be inside the reactor, but high compared to normal. Favors. Oh, here it is. The environment does what? It favors. It favors adaptation to oxidative stress in birds. Well, you would think that these birds would be a bunch of mutants because their DNA would be blasted with all these radioactive particles, but they're not. The target is ionizing radiation shown by molecular damage. As far as we know, they don't have a sensor for the radiation, but they have sensors in their body for tissues that have been damaged by radiation. In fact, they're called reactive oxygen species. Now, when these things get hit, the body is able to detect those. It detects the target of the radiation. And guess what? A signal is sent to their DNA processing area. It upregulates DNA repair mechanisms across the board. So once it sensed that, the DNA which is actually getting damaged by the radiation is rapidly being fixed and repaired across the board. So the paper says, thus, when several species are considered, the overall pattern indicates that birds are not negatively affected by chronic exposure to radiation and may even obtain a beneficial homeotic effect following an adaptive response. Meaning this, they upregulate all DNA repair, so they don't live shorter lives, they actually live longer lives to birds who aren't exposed to it, who haven't upregulated their DNA repair. Wow. I suspect this ability has been in those birds from the beginning. And it is an ability which probably, chronic radiation exposure probably wasn't on the earth at the time. So it is an ability which is not due to the chronic radiation exposure. It is an ability which preceded the chronic radiation exposure. Do you see what I'm saying? That didn't cause it. It was already there. Wow, those are some epigenetic changes. Let's look at a few more interesting changes. This is a study on these little fish that live in some Mexican streams, mechanisms underlying adaptation to life in hydrogen sulfide-rich environments. Well, these fish live in streams that have been polluted by hydrogen sulfide. And guess what? They have chemosensors for hydrogen sulfide. And where do you think the sensors are on their body? They're actually in their gills. So as that water flows through the gills, they detect hydrogen sulfide, and they adjust themselves to live in this. In fact, how do they adjust themselves? This is illustrative in a lot of ways. They upregulate, on average, over 1,600 genes, and they downregulate, on average, over 1,800 genes. This is an across-the-board change to that. Now, many of people always think, well, what is the gene for this, or what is the gene for that? Well, we don't generally have a gene for anything. Most genes are multifactorial. Multifactorial. And so this is hundreds of genes that are being upregulated, and they're specific for hydrogen sulfide detoxification. As it says there, it's not that they are necessarily turning on some other unrelated genes. It's really that the genes that have been previously implicated in hydrogen sulfide detoxification are turned on or turned up. And the ones that are turned down are genes that would metabolize hydrogen sulfide into something even more toxic. So they are able to fit this environment with hydrogen sulfide. We talked about the blind cave fish. The usual story is that they find themselves in a cave and then the eyes become a detriment, and through selection, wiping out those with eyes, it eventually selects for those with eyes, without eyes. However, look at the fish. Not only does it not have eyes, it is also hypopigmented. Is it not? And I read a paper that said the space in the skull that the eyes used to fill, it doesn't just stay as an open void. Portions of the brain actually develop and will fill that void, and the portions of the brain are for sensors for touch and smell and other things which work in a dark environment. So I was thinking, wow, you lose your eyes, you lose your pigmentation, and you change other parts of your brain enabling you to fit in the dark. This sounds like this is a design system enabling fish to live in these caves. So I wrote an article saying, what if the typical creationist and evolutionary explanation is totally wrong, of which the creationist is just the same thing as the evolutionist, only they don't extrapolate it as far? What if there's a mechanism which allows these fish to move in a cave? And sure enough, there's a target which are abiotic factors in the water, and these fish have sensors for those. In this particular case, it's not light and dark, it's for water conductivity, which is different in cave water versus stream water. And the paper asks a very important question. A critical question is whether a fish finding itself suddenly trapped in a cave environment would experience a heat shock protein 90 related stress response. In other words, would it lose its eyes, pigmentation, and the other changes that I just discussed? And the answer is yes. In one generation, as I mentioned yesterday, depending on what environment these fish grew in as tiny little embryos. They had no eyes to partial eyes to fully eyed in one generation. And this paper came out about about 14 months after I published that article on Axum Facts. Now, I'm not saying that to puff myself up. I'm just saying that when you have the proper thinking, you can actually make predictions. And when people say creationists don't make predictions, here's a published prediction which came out 14 months before the paper came out. So creationists did make a prediction of what was going to be found based on this idea of continuous environmental tracking. Okay, look at this little lizard. This lizard lives on an island off the coast of Greece. It was transplanted there about 40 years prior to that, and it lived in another space around Greece where it ate mostly insects. But when it was transplanted to this island, there were not the insects that it ate, but there were plant materials. And so somewhere in its gut, it has a sensor for plant materials and certain types of microbes, particularly for high cellulose plant material. Because the lizard didn't die just because it couldn't eat its insects. It started eating plants. Well, when you take a lot of plants into your gut, if you want to get the nutrition, they actually have to be broken down and fermented by the bacteria in the gut, which are going to liberate the nutrients. So guess what? This lizard developed a whole new structure in its gut, a cecal valve, and a tiny little cecum, which it did not have, which shuts off the flow of food through the gut, holds it in a pouch, allows the bacteria to break it down. It absorbs the nutrients. And these lizards are genetically identical to their relatives, which are still eating the insects. So there was no genetic change. Nothing was changed. It had the ability all along, when it needed to make a cecum and a cecal valve. And guess what? When you take them out, it can disappear. It's not due to that. It had the ability to be able to be fruitful, multiply, and what? Fill. Even before that ever happened. And this just shows they're genetically indistinguishable from their cousins. Well, these are plant hoppers. Plant hoppers live in all different types of environments. There's an insulin receptor, which is really an insulin detector, which adjusts the length of their wings and several other factors on them. The target is population density and nutrient quality, and nobody knows exactly what the sensors are, but I'll kind of give you an idea what I think they are. On grasshoppers, there's a tiny little sensor on the back of its rear leg. And when other grasshoppers touch that sensor, touch it, and when it gets touched a certain number of times, the grasshopper morphs into a locust. And then it flies away and populates a new area. So in other words, when the population of grasshoppers gets too high, which is determined by how many times it gets bumped on the rear leg by other grasshoppers, it completely changes its shape and flies to a new location. That's probably what happens with these plant hoppers as well. Plus, they're able to detect the quality of the food they're getting, so if it goes down, they change. So after that happens, you can have two different morphologies of the plant hopper. A short-winged one, which doesn't fly but has high fertility, and a long-winged one, which can fly but has lower fertility. So depending on its nutrients and depending on its population density, the shape of the bug completely changes, allowing it to be fruitful, multiply, and fill new environments that are not overpopulated with plant hoppers. Wow, that's interesting. And then the final one is this tiny microscopic worm called a nematode. You can't really see him. This is an electron microscopic photograph of it. Now these nematodes, they generally live on fig plants. But they don't just get to the fig plants by migrating. They're carried to the fig plants by a wasp vector, which eats the nematode, and the nematode passes through the wasp, and the wasp lays it on the figs. Now these little nematodes, they're able to target the fig maturation time. And they actually have sensors all over them for the types of figs, the fig phase, and whether they've actually passed through the wasp or not. So they can tell all of that. And once they pass through that and they go to various different figs, this is what's incredible. Their developmental plasticity, in other words, their built-in mechanisms, allow discontinuous novelties from a single genotype. And they produced 5 different shapes, which were so different from each other that not only would you have not thought they were the same species, you wouldn't have even thought they were the same genus. In fact, some of them have structures with no analogs in any other nematode. In other words, they are morphologically distinct. They're morphologically distinct from all the other nematodes, but they are genetically identical to all of them. You see what I'm saying? You get major changes from the same genetics. It's all on how it is regulated. And that's allowing these nematodes, even tiny little nematodes, to be what? Fruitful, multiply, and fill the earth. And this paper said, macroevolutionary scale. You know what they mean by macro? They mean one type of creature to another type of creature. Macroevolutionary scale diversification without genetic divergence, giving the empty niche conditions. Be fruitful, multiply, and what? Fill the earth, even these niches, predisposing an ecosystem to the trophic diversification and its colonists. So you have massive, wide-scale diversification, and we covered all of these examples. Did I tell you that the Lord is just very creative? How many of these involve death and struggle? None. None. All of these mechanisms, and believe me, many, 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 many more to be discovered and many more to just be elaborated on, these were probably all operating from the moment these creatures were created, before the fall, before there was ever death and struggle, before there was so-called survival of the fittest. These mechanisms were allowing organisms to be fruitful, multiply, and fill the earth from the beginning of creation. And guess what? They weren't ruined by the fall. The fall came and they just kept working away. Just kept working away. So why, given these elaborate mechanisms, would anybody ever want to embrace the evolutionary mechanism of what? Survival of the fittest, which is, it's bad in every way. First of all, it's idolatrous, which you now know. Second, no engineer would use it because it's trial and error. Trial and error, trial and error, trial and error. Maybe some will work, maybe some won't work. So it's very, very inefficient on top of all of that. And then you just import evolutionary thinking into your worldview. We as creationists should embrace this new research, frame it ourselves afresh and anew, guided by biblical principles that sees the handiwork of the Lord and shows how organisms were made to continuously track their environments, sometimes very rapidly, sometimes over multi-generations, sometimes with epigenetic changes to rapidly flex when the environment quickly changes. And oh, by the way, when the environment goes back, most of those epigenetic changes go away and the organisms revert back to where they were. Whoop, whoop. I mean, all of this is just phenomenal genius and good design. And we should be the leaders in promoting this and getting it out. It will really honor the Lord. Dr. Nicholas, thank you so much.